Genetics helps us to understand the causes of biological variation. And so, we're not really going to talk about genetics. It's much more complicated than what you learned in high school biology. Um, our genetic material is made of DNA molecules, deoxyribonucleic acid molecules. These make up genes and chromosomes that are our basic hereditary units. When we look at kind of how we can inherit variation, there are biochemical changes or mutations in DNA that provide much of the variety on which natural selection operates. So if you recall back to high school biology, you remember that we've got four base pairs, adenine, guanine, cytosine, and tyrosine. There's also a U. I'm spacing on what that one is, that's present in RNA, but not in DNA. You remember the stages of mitosis and meiosis, right? The chromosomes condense. They line up in the center of the cell. They're joined at the centromere. In mitosis, the copies of the chromosomes split apart, and each new cell has all 46, if in the human case, chromosomes. In meiosis, as they split apart, they only get half, so they don't double before or replicate before splitting in half, and so the gametes then each have 23 chromosomes. It's Gregor Mendel who first began to understand kind of basic, what we call Mendelian genetics. And he was an Austrian monk and really chose the perfect plant to work with. He chose pea plants, and like to spend a lot of time in the garden. And so he looked at firstly 7 contrasting traits in pea plants and discovered that heredity is determined by discrete particles or units. And as he kind of moved forward in his generations of peas, he was able to make predictions about what kinds of offspring would be present. Now, the reason I say that Mendel got lucky is that he chose traits that are what we call one gene, one trait, which means that variation in the alleles at one location, at one gene, variation in the outward physical appearance. And so he looked at things like red flowers and white flowers. He looked at yellow peas versus green peas, at wrinkled skins on the peas versus smooth skins on the peas, at inflated pods versus kind of dried out and shrivelly pods, so on and so forth. And he found that when he crossed what we would consider pure parents for one trait, they would be homozygous for one trait. That means they possess 2 of the same alleles. In the first generation, they all express the dominant characteristic or the dominant appearance. So in this example on this slide, we've got red flowers and white flowers. The red flowers are dominant to white flowers. That means in this first generation, the F1 generation, all of the offspring will produce red flowers. Where it gets interesting is in the second generation, where you get this ratio of 3 to 1, 3 red flowers to 1 white flower. When we explain this, we talk about these kind of discrete, the trait, let's say, this discrete unit. So these red flowers have the alleles big C, big C. The white flowers have these alleles little c, little c. When the first generation is crossed, you end up with all of the offspring as heterozygotes, meaning they've got 1 big C inherited from the mother and 1 little c inherited from the father. Then when you cross heterozygotes, you get an appearance ratio of 3 to 1, but you get a genetic ratio of 1 to 2 to 1. So you've got 1 offspring who is big C, pure homozygote for red flowers. 1 offspring who's big C, or 2 offspring who are big C, little c, heterozygotes for red flowers. And 1 offspring that's little c, a homozygote for white flowers. And he went on and crossed, comparing multiple traits at a time, and found that even as you crossed looking at 2 traits at once, that you got these same kinds of ratios. This explanatory kind of hypothesis here, this is where we talked about Punnett squares when we were in high school biology. Mendel concluded that dominant forms could mask recessive forms in these hybrid heterozygote individuals without destroying the recessive trait. And so these genetic units that Mendel described were factors that we now call genes. The variation in the kind of outward appearance. So the gene would be flower color. The allele would be red or white. And these are located on chromosomes. Chromosomes are paired lengths of DNA that are comprised of multiple genes. A gene is a place on a chromosome that codes for or determines a particular trait. Alleles are biochemical variants of particular genes. So let's think about blood type. Blood type's a nice one to study in humans. We have one location for blood type, one gene for blood type. That gene has three alleles. Those alleles are A, B, and O. Two of those alleles are codominant. That's A and B. One of those alleles is recessive. That's O. So when we talk about possible genotypes, you can have type O blood, where you have two recessive alleles, from both parents. And that's you're then homozygous for O. You can have type A blood and be either homozygous for A as the allele. That means both your mom and dad contributed type A to you. Or you can have type A blood and have an A allele and an O allele because that O is recessive. Same holds true for type B blood. You can be homozygous for B and have two B alleles, or you can be heterozygous and have a B and an O allele. And then in a case where you've got type AB blood, both of those are dominant and both of those are expressed. So, our blood types are a good kind of example of how alleles can then determine our different phenotypes. And so this here is looking at how we can have independent assortment and linkage. Generally, we expect that genes are going to independently assort. That means they're not going to travel together. They are going to, particularly if they're on different chromosomes, have an equal probability of being present in each of the gametes. There are some factors that can lead to differential heredity, including linkage. If they're on the same chromosome, then they're much more likely to travel together versus breaking apart, unless we have crossing over where the ends of the chromosomes switch little bits. We're not going to test on any of this bit. This is just so you have this basic understanding what we mean when we talk about genetic evidence for things, the evolution of certain genetic traits. So some of the key vocabulary words that come from Mendel's experiments include heterozygous. This is where you have two different alleles, like having an A and an O allele, but still having type A blood. Homozygous. This means that you have identical alleles of a given gene. So this for type A blood would be having two A alleles. This is also a point where we need to talk about the difference between genotype and phenotype. Your genotype is your hereditary makeup. Your phenotype is the observed expression of the trait. So with blood types, if you have type AB blood, your genotype and phenotype are the same, right? You've got an A allele and a B allele, and your blood type is thusly AB. Whereas with type A blood, you don't have a perfect matchup between your genotype and phenotype. You can have type A blood with two A alleles, or you can have type A blood but still have an O allele. So dominance can produce a distinction between genotype and phenotype. One thing to keep in the back of your mind is that we measure evolutionary change through changes in gene frequencies over time within a population. And so we are measuring at the population level changes in genotypes over time. That is our kind of end point. And that allows us to say that evolution has happened and here's what we've measured. Natural selection, though, operates on phenotypes. It's not considering genotypes when the environment is culling certain members from the population. If all of a sudden we saw the emergence of a new virus that only attacked people who had type A blood, it's going to weed out some O alleles from the population because the hosts that have those O alleles in this heterozygous state are going to die. So the environment acts with upon phenotypes to select those that are either going to survive or reproduce in higher numbers. We also have to talk about sources of genetic variation. Everybody harps on mutation. Yes, mutation is important. Mutation arises by the imperfect copying of DNA strands. In humans, our mutation rate is 10 to the minus 4 to 10 to the minus 6 per gene per generation. We've got about 30,000 genes. That means we get about one mutation per every 3 offspring born. Well, why aren't we X-Men, right? Why don't we have the rise of quote unquote mutants? Well, there's a lot of redundancy in our genetic code. And so if you recall back to high school biology, the DNA is copied by RNA in units called codons. That's 3 base pairs together. Those codons are interpreted by the ribosomes to generate amino acids. Amino acids, therefore, are kind of coded for or determined by this collection of 3 base pairs. There's some redundancy in the way that codons result or translate into amino acids, in that you can change oftentimes the middle base pair and not change the amino acid. So you could have a mutation that changes one base pair in your DNA that doesn't change the amino acid that's produced, which then doesn't change the protein that forms from a string of amino acids. We also have some redundancy in the way that amino acids form proteins. So you can change one amino acid because, let's say you change the last base pair, and you don't change the functional protein because changing that one amino acid doesn't change how the protein folds in upon itself and its kind of finished structure. So a lot of the mutations that we experience are functionally neutral. So, we're not getting rise or giving rise to individuals who can apparate or control the weather or have laser eyes or anything like that, right? Most of our offspring who are born with mutations, those mutations don't do anything. Also, even if they do something, it doesn't mean that it's going to visibly change anything about our offspring. So that mutation might code for something, but it's not going to change the physiological response that our organ systems have when presented with certain environmental stimulus. What's more interesting with evolution is sexual recombination. This happens every time we reproduce. And when you think about it, for humans, for mammals, for birds, sexual recombination is the only way that you get offspring. You're not able to, for example, clone yourself. So there's some benefit that comes with sexual recombination that paid off pretty early in our evolutionary history. So sexual recombination is defined as the production of offspring with a combination of traits that differ from those found in either parent. It is this critical payoff to sexual recombination comes in our evolutionary arms race with pathogens. So, think about it. When we look at plants, they have the ability to both reproduce asexually through cuttings or through a sprout coming up from a root, etc., and sexually through the production of pollen and flowers that then receive that pollen. When we think about bacteria, they generally reproduce asexually. They cleave and kind of produce 2 daughter clones, but they also engage in something called cytoplasmic bridging, where a connection will come between 2 different bacteria, a little cytoplasmic bridge that forms, and DNA will then be exchanged. So even as far back evolutionarily as bacteria, we've got some benefit to getting new and novel genetic combinations. Fish can sometimes reproduce asexually through parthenogenesis, where they lay eggs that haven't been fertilized, and can sometimes reproduce sexually when they spawn with males. This demonstrates kind of this sticking point or benefit in this evolutionary arms race. There's a species of fish, I can't recall the name, but they're found in the Chiricahua Mountains of Arizona, southwestern New Mexico, and the northern part of Mexico, right there across the border. And as desert fish, their life cycle is kind of, it depends upon what's happening weather-wise. So during much of the year, the washes or kind of the riverbeds that these fish live in dry up to where you only have isolated pools. And during some periods of the year, like the monsoon rains, there's flooding and these pools mix. What's interesting about these fish is that they're infected with a parasite, and that parasite shows up by basically just a black dot on their skin. So you can literally look at these fish and count the number of black dots they have and know how many parasites they have. What we find is that when the pools have dried up, or when the rivers dried up and you've just got these isolated pools, you've got a pretty consistent environment. And in that case, the asexual reproducers do best, those that lay unfertilized eggs that are clones of the mothers. Those individuals have fewer black spots, therefore fewer parasites. However, during the monsoon season, when there's flooding and mixing of these populations, the sexual reproducers do better. They've got fewer parasites. So what this proposes is that during times when the environment is not changing, if you're well suited to the environment, it might be worth it to reproduce asexually. However, when the environment is changing, there's a benefit to sexual recombination because you don't know what genetic combination is going to give the best immunity to your future offspring. And so you try to find a mate with whom you can produce a variable offspring that hopefully one of them will have that genetic combination, that immunity that's going to pay off in the long run. And so with respect to our evolutionary arms race with microbes, we don't know the microbes that we're going to be faced with 20 years down the road, 30 years down the road, 100 years down the road. And so we're looking for new and novel immune systems. We are using cues. They're olfactory. That means they're smell-based cues. We smell pheromones of people. We detect their immunocompetence through their body odor. I mean, if you've ever been in a long-distance relationship, you've got their sweatshirt of your long-distance partner, and you bring it up to your nose, and you sniff, and you're like, and it brings you peace and comfort, a release of serotonin, of dopamine, of oxytocin, all those feel-good things that make you happy and kind of reinforce that bondedness. If your friend smelled your partner's sweatshirt, they might be turned off by the odor because it's not a good immunological match for them. So, the nose knows. The nose is able to pick up on some of those cues that allow us to determine whether someone is a good genetic match. One other form of variation is random genetic drift. These are random changes in gene frequencies that appear due to chance. So the best way to exemplify this is I've got a bag. Okay, we're going to play a little game. And so you think of these questions and answer them in your own mind. I've got a bag that's got 5 red marbles and 5 blue marbles. I'm going to ask you to pick 5 marbles. All right, you probably picked 3 to 2, a ratio. It might be 3 blue, 3 and 2 red, or 3 red and 2 blue. I'm going to say we pick 3 blue and 2 red. I'm going to double that. That's our next generation. So now I've got a bag. It's got 6 blue marbles and 4 red marbles. Again, you have to pick 5. Well, from probability theory, you've got a higher likelihood of maybe picking 4 blue marbles and 1 red marble. I'm going to double that for the next generation. Now we've got a bag that has 8 blue marbles and 2 red marbles. Now there's a chance when you pick 5 marbles, you're going to end up with 5 blue ones. In just 3 generations, we've gone to fixation for blue as the color of our marbles. This is, for example, what happened with blue eyes reaching fixation in Scandinavia. It was a founding effect. There was a population, and by chance alone, there were more people who had blue eyes than brown eyes who founded the Scandinavian populations. And so these traits then went to fixation. Now, people who have Scandinavian heritage are much more likely to be blonde-haired and blue-eyed.